Good afternoon, dear students. The topic of the lecture, molecular basis of heredity. Plan lecture. At first, development of the notions of molecular mechanisms of inheritance. Secondly, chemical organization of genetic material. DNA structure, properties and functions. And the next one, nuclear and mitochondrial DNA structure features. RNA, types, structure, biological role. The next one, self-reproduction of heredity material. Principles and stages of DNA reduplication. Features of reduplication of mitochondrial DNA. At the last lecture, we found out that the basic level of living organization is the molecular genetic level. And the basic molecule of heredity is the DNA. The opening DNA structure let understand how the cells and organisms are reproduce themselves, how the necessary for life information is encoded in them. In the early 20th century, Seton and Boveri, independently of each other, detected that transmission of hereditary information happens through chromosomes. Griffiths, Avery, McCarthy and MacLeod located the role of the DNA, not protein, in the transmission of hereditary information. Hershey and Chase proved universality of DNA as a carrier of hereditary information. Earlier thought that genetic information passed by the proteins. Linus Pauling studied the alpha helix structure of proteins, and it helped for studies DNA structure. At the same time, Maurice Wilkins, physicists, and Rosalinda Franklin, molecular biologists, did X-ray diffraction analysis of DNA. By this time, biochemistry's Chargaff formulated rules of complementarity. 1. Adenine is equal to thymine, and guanine is equal to cytosine. 2. The number of purines is equal to number of pyrimidines. 3. The number of bases containing amino group at position 4 of pyrimidine and at position 6 of purine nuclei is equal to the number of bases containing oxo group at the same position. The scientists of Cambridge University Laboratory, James Watson and Francis Crick, tried to build special models of DNA. In 1953, they published the data of DNA structure and were awarded Nobel Prize. Let's consider the structure of nucleic acids. The nucleic acids are macromolecules, those molecules with high molecular weight. Nucleic acid is polynucleotide, which consists of monomers, are nucleotides. A nucleotide molecule consists of 3 parts: a pentose, the nitrogen bases, and phosphoric acid. Pentose is represented by ribose in RNA and desoxyribose in DNA. The nitrogen bases are purine, adenine, and guanine, and pyrimidine, cytosine, and thymine or uracil. The nitrogen in the rings gives the molecules basic properties. Phosphoric acid gives the nucleic acid acidic properties. Then the first carbon atom of pentose connects with nitrogen base from the nucleoside. This connection is condensation reaction. A nucleotide molecule forms after the second same reactions. At the same time, nucleoside and phosphoric acid connect each other through a phosphoester bond. Two nucleotides form dinucleotide with the help of the phosphodiester bond between phosphate group of one nucleotide and hydroxyl group of another. These bonds give to nucleic acids strength and stability. Then the synthesis of polynucleotide is go on. The process repeats again and again, to several million times. As you know, there are DNA and RNA. Let's remember the structure of DNA. The DNA molecule consists of two antiparallel polynucleotide chains, which swirl to the right. The antiparallelity of polynucleotide chains is provided by linkage of 5' end one chain to 3' end or the other. Two chains are bonded to each other with help of hydrogen bonds between bases. The pairs linkage is very specific. There is only two possible pairs, adenine and thymine, and guanine and cytosine. The sequence of pairs in one chain may vary in wide range, but the sequence of pairs in the second chain has to be complementary to it. Thus, the pair sequence in one chain defines the complementary sequence in the other chain. So, the DNA structures are: primary polynucleotide chain, the basic bond is a phosphodiester bond. And the next one, secondary structure. Two complementary to each other antiparallel polynucleotide chains. The basic bonds are hydrogen bonds. And the next one, tertiary structure of DNA. Three-dimensional spiral with characteristics described above. The DNA is superspiral, which can compact. If we take all human DNA molecules from all chromosomes of one cell and put them in a line, its length will be about 2 meters. The total length of all DNA molecules from all cells of the human body will be 10 to the 11th power kilometers. This is almost 700 times the distance from Earth to the Sun. There are two types in cell genome. At first, nuclear genome, 60 to 70,000 genes, or 19–50% of genes, and mitochondrial genome, only 5% of all genes. The nuclear DNA structure we know now. Let's speak about the mitochondrial DNA. The mitochondrial DNA localized in mitochondria. The mid genes are plasmogenes. The plasmogenes form plasmon. This is said non-nuclear genetic material which inherited on the mother's side. So the DNA structure includes the 37 genes. 13 encodes the synthesis of oxidative phosphorylation proteins. 22 encodes tRNA synthesis, and 2 synthesis of rRNA. The tRNA and rRNA genes, evidence of a proprietary apparatus for protein synthesis in mitochondria. The structure of RNA. The RNA molecule is single-strand. It consists of such nucleotides as adenine, guanine, cytosine, and uracil instead of thymine. There are the following RNA types: pro-matrix or pre-messenger RNA, matrix or messenger RNA, transfer RNA, and ribosomal RNA. Matrix or messenger RNA. mRNA contains 3 to 5% of the total RNA in the cell. It is formed in the one of DNA strands in the stage of processing during transcription. The base sequence in mRNA is a complementary copy of the DNA matrix strand. Before mRNA synthesis forms pro-matrix or pre-messenger RNA. It consists from exons and introns. During the processing, exons are deleted and introns connect, so mRNA forms. The shortest mRNA molecule consists of 300 nucleotides. Most mRNAs exist in the cell for a short time. And the next one, ribosome RNA. rRNA is up to 80% of the total RNA of the cell. The rRNA base sequence is similar in all organisms, from bacteria to higher animals. rRNA is also contained in the cytoplasm, where it is linked to proteins, forming cellular organelles, ribosomes or polysomes. And the next one, transfer RNA. tRNA delivers the amino acids contained in the cytoplasm to the ribosomes. This RNA has a short nucleotide chain of 80 to 100 nucleotides. tRNA is like a clover leaf, an acceptor stem formed by two complementary connected ends parts. The first part of tRNA consists of 7 base pairs. 3' end of this stem is longer. It forms a single-strand section that ends in a CCA sequence with a 3 or H group. The transported amino acid is attached to this end. Another 3 parts are 1. Anticodon. The sequence of bases in the triplet of the anticodon strictly corresponds to the amino acid trait these tRNA carriers. 2. Dihydrouracil. And 3. Pseudouracil. Go to the next point of the lecture, the DNA reduplication. I believe you know the DNA is only one molecule that can reproduce itself. This property is the basis for storing and transmitting inherited information. This is the DNA reduplication. The DNA reduplication is the ability to copy itself. In the process of reduplication, a complementary chain is synthesized on each parent polynucleotide chain of DNA. The result is 2 additional spirals. Each daughter DNA contains one parent and one synthesized chain of polynucleotides. This replication is called semi-conservative. Okay, let's watch the video and then take a closer look at the process.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Well, phases of DNA reduplication. Initiation, beginning, elongation, build-up, extension of non-building daughter macromolecule of DNA. Termination, ending. Phase of initiation. In the G1 period of the cell cycle, a pre-replicative complex of 15-20 different proteins is formed. There are initiating of recognizing proteins and destabilizing proteins. Initiating proteins find the initiation points of DNA molecule. The double-stranded DNA molecule separates at one end with the help of helicase enzyme. The regions of DNA despiralizing by helicase enzyme are called replication forks. The DNA chains are held at distance each other using destabilizing proteins. Each parent chain becomes a matrix for new complementary chain synthesis. At first, an enzyme called the primase starts the process. This enzyme makes a small piece of RNA called a primer. This mark is starting point for the construction of a new strand of DNA. The DNA fragment from the point of replication starts to the point of replication and forms a replication unit, a replicon. The eukaryotic cells have a large number of replicons. That's how the replication of DNA of eukaryotic chromosomes starts at several points. The next phase is elongation. This is extension of daughter chains of DNA molecule. As a result of this, from one DNA strand appear two with the same structure. The process happens with help DNA polymerase enzyme. Types of DNA polymerase are alpha DNA polymerase. Delta DNA polymerase provides direct DNA replication. Beta and epsilon DNA polymerases are involved in repair of DNA molecular damage. And gamma DNA polymerase. provides mitochondrial replication. During a replication process, the replication fork moves along mother spiral. The ability of DNA polymerase to add nucleotides only in direction from 3' to 5' means that the process of replication in two DNA strands should be different. On a one matrix, the replication of DNA occurs continuously from 3' end to 5' end. On another matrix, the process of replication performed by short fragments. Then the short fragment of DNA are added to the growing chain. In the 3' to 5' direction, DNA polymerase jumps ahead to fill another gap. These fragments are called Okazaki fragments, consist of 100 to 200 nucleotides. The synthesis of each fragment is preceded by the formation of an RNA primer, about 10 nucleotides. Then they connect by DNA ligase enzyme. The first strand is called leading strand, and the second strand is called lagging strand. The ending phase is termination. The replication process in eukaryotic cells stops when two replication forks of two adjacent replicons meet. Adjacent fragments are ligated to each other under the action of the DNA ligase enzyme. Now we watch the video again, but you will try to determine the phases of DNA reduplication yourself. Mitochondrial DNA has the specific features of reduplication. At first, mitochondrial DNA replication is not related to the cell cycle, interphase period S, and occurs at any of its time points, except for mitosis itself. Secondly, the mitochondrial DNA of individual organelles replicates independently of the DNA replication of other mitochondrial cells. Over the cell cycle of DNA, some organelles replicate more than once, and others do not replicate at all. And the next one, replication of mitochondrial DNA is catalyzed by enzyme gamma DNA polymerase. And the next one, formation of RNA primers in replication, mitochondrial DNA catalyzed DNA-dependent enzyme RNA polymerase. When the replication fork reaches the end of a linear eukaryotic DNA molecule, on which the lagging daughter DNA strand replicates like a template, there is no room for the formation of an RNA primer, from which DNA synthesis or the Okazaki terminal fragment could begin. As a result, with each replication cycle, DNA molecules are shortened. In human body, by 50 to 100 nucleotides. This is the DNA marginotomy and mechanisms of prevention of shortening or marginotomy of DNA. At first, telomere DNA is formed by nucleotide repetitions reaching guanyl nucleotide. And secondly, special nucleotide sequences are recognized by enzyme telomerase complex with reverse transcriptase properties that extends the corresponding terminal DNA fragments on its RNA matrix. The level of reliability of DNA replication in eukaryotic cells is quite high and amounts to 1 error per 10 to the 9th to 10 to the 10th power nucleotide pairs. This ensures reliable storage and accurate transmission of hereditary information from generation to generation by DNA molecules that are the basis of all living beings. Thank you for your attention.